Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, we are in a continuation of the chapter that is molecular basis of inheritance. And in that, in previous class, we have initiated a topic that is transcription. And in that, we have discussed already about the transcription unit and the different types of RNA. Today, we are going to actually the mechanism of transcription in case of the prokaryote. And first, we are going to talk about the mechanism of transcription in a prokaryote, that is bacteria. If we talk about transcription, it is a process of formation of mRNA from the DNA. And obviously, to carry out this process, there is a need of enzyme. And here, RNA is going to synthesize, which is a polymer of a ribonucleotide. So, RNA polymerase enzyme is getting used. But here, the enzyme is working based on the dependency of DNA and synthesizing complementary sequence to the template strand. So it is DNA dependent RNA polymerase which is going to form a chain of ribonucleotide as RNA, and that is single. Only one enzyme is used that catalyzes transcription of all types of RNA in bacteria. Okay, all types of RNA in bacteria are transcribed by only a single type of RNA polymerase. Whereas in eukaryote, we will study that there is a different types of RNA polymerases are involved. Now, what RNA polymerase do? It bind to the promoter and initiate the process of transcription, or we can call a initiation. It uses nucleoside triphosphate as substrate, or we can say nucleotide as a substrate, and polymerizes in a template dependent fashion. Means it uses a 3' to 5' strand of a transcription unit as a template, and complementary to that mRNA is getting synthesized. Now, if we talk, then this transcription in prokaryotes, transcription in prokaryotes occur in cytoplasm, occur in cytoplasm of cell. And here, the transcription enzyme is of only one type, that is a DNA-dependent RNA polymerase. And that synthesizes all type of RNA, that is either mRNA, tRNA, or RNA. And all three RNAs are needed for synthesis of a protein in a cell, that is mRNA, tRNA, and RNA. Now, if we talk about this RNA polymerase, this RNA polymerase is a holoenzyme, is a holoenzyme. And if this RNA polymerase is a holoenzyme, it is made up of two things. It made up of it is made up of sigma factor and core enzyme. Now this core enzyme is made up of alpha 2, beta, beta dash, and omega factor. Sigma factor, alpha 2, beta, beta dash, and omega factor. This all are the different polypeptides. So the sigma factor is a polypeptide, alpha 2 is also polypeptide, beta, beta dash, and omega all are the different polypeptides. But they combinedly form a one holoenzyme which is made up of these two parts. So the enzyme holoenzyme without the sigma factor is known as core enzyme. Now this whole process of transcription in prokaryotes occur in three phases. Okay. So here process of transcription completes in three steps. Complete in three steps. First, initiation. Second, elongation. And third, termination. In these three process, whole process take place. Now, how this happens? Let's study. Now, if you want to understand the initiation is a sigma factor dependent. So, suppose this is a DNA strand, and this is in 3 dash to 5 dash direction. Now, another strand is also there, that is in 5 dash to 3 dash direction. Now, both between both strands there is a complementarity. And between both strands there is a complementarity. So this is a DNA which is double stranded. Now here, suppose we can say this is the promoter region, and suppose this is the terminator region. Now what will happen? First, holoenzyme bind with the promoter sequence and initiate the transcription. Holoenzyme bind with promoter sequence and initiate the transcription. And when it initiate the transcription, the whole holoenzyme is involved. So here the region from where the transcription initiates, where a unwinding or a breakdown of hydrogen bond takes place. And when the breakdown of hydrogen bond take place, the two strands getting separated. And here the whole holoenzyme binds alpha 2, beta, beta dash, omega, and sigma factor. Whole holoenzyme binds and add first ribonucleotides. Okay, and this is the short stretch of RNA which is synthesized, and this is a initiation. Now further breakdown of hydrogen bond and unwinding continues further, and this holoenzyme moves further and elongate. And when elongation take place, the diagram somewhat and this is the particular DNA. Now this is the elongating phase, and during elongation the RNA strand is getting synthesized. Okay, but be remember here only alpha 2 beta beta dash omega is involved, because sigma get factor get released after the initiation, and sigma factor is required only for initiation of transcription. After that, core enzyme will carry out a elongation, and at last the termination will occur. So now after elongation, the termination will occur, that we will draw in the next slide. So when termination take place, this is 3 dash to 5 dash, and this is 5 dash to 3 dash. So this is 3 dash to 5 dash strand, and this is 5 dash to 3 dash strand, and here at the terminatory region, when core enzyme reach, it bind with the rho factor, and rho factor carry out a termination of the mRNA. So mRNA get separated. So this is a termination. So termination occur of mRNA when the rho factor bind with the core enzyme. So termination is rho factor dependent. Termination is rho factor dependent. Okay. So such a way the whole transcription is going to occur, and an mRNA and get synthesized in case of the prokaryote. So in prokaryote, in prokaryotic transcription, initiation is occurred by a holoenzyme, so it is a sigma factor dependent, whereas a particular termination is rho factor vector. So RNA polymerase bind to the promoter region during the initiation of the DNA, and the process of transcription begin. It is utilizes nucleotide triphosphate as a substrate, and polymerizes in a template dependent fashion following the rule of complementarity. It also helping opening the helix and continues elongation. So helix is opened also by the particularly holoenzyme. And initially only short stretch of RNA attached to the enzyme. Once the polymerase reaches the terminal region, the nascent RNA or RNA polymerase falls off, or it is released from the DNA helix, and it result in termination of the transcription. Okay, so such a way that particularly transcription take place in case of the prokaryote. Now, some points are getting understood by the particularly this prokaryotic transcription. So in prokaryotic transcription, if we talk, then number one, mRNA does not require any processing to become active, and it does not require any processing to become active. So the ultimately fall of mRNA or a newly formed mRNA is the matured one. Second point, here there is no between bacteria, there is no membrane bound organelle. So transcription and translation means protein synthesis, mRNA synthesis and protein synthesis take place in same compartment. In same compartment, that is cytoplasm, and there is no separation of, and there is no separation of cytosol and nucleus. And third thing, many time the translation begin much before begin much before the mRNA is fully transcribed, much before the mRNA is fully transcribed. Thus, we can say transcription and translation can be coupled in bacteria. So if we talk about this point, and if we want to understand that mRNA is a mature one, there is no need of processing, and both transcription and translation occur in same compartment, and translation and transcription are coupled. So what do you mean by coupled? Suppose here mRNA is getting formed, and ribosome ultimately attached to this, and ribosome start making protein. So if this ribosome start making protein without the completely termination of this transcription, then we can say the transcription starts before the completion of the translation starts before the completion of this transcription, and that way, at that time we can call it as a what, a transcription and translation are coupled. Okay, dear students, I hope you understood it in very well manner, and that is all about the water transcription in case of the prokaryote. So these points are important. You should note down it. These are important points. Okay, and that help us to differentiate the prokaryotic transcription from the eukaryote. So it somehow facilitate the opening of the helix and continues elongation. Elongation continues by the opening of the helix in the front part of the bubble, which is formed by the breakdown of hydrogen bond. Only a short stretch of RNA remains bound to the enzyme, obviously, and other formed RNA get come off the double helix. Once the polymerase reaches the terminal region, the adjacent RNA falls off. So also the RNA polymerase that also get released, and this result in termination of transcription. The RNA polymerase is only capable of catalyzing the process of elongation. Okay, and that's the structure of what RNA polymerase, which is used in the transcription in case of the prokaryote. It associates transiently with the initiation factor, sigma factor, okay, and termination factor to initiate and terminate the transcription respectively. Means transient means for a short period. Sigma factor attached to the core enzyme only for the initiation, and the rho factor attached with the core enzyme for the termination. Association with this factor also the specificity of the RNA polymerase to either initiate or terminate. This is the specificity from where the need to initiate and from where it to had to terminate, that is specified by RNA polymerase by using these factors. In bacteria, the mRNA does not require any processing to become active. It also since transcription and translation same compartment and they are coupled that we have discussed. Okay, now after this, we are going to talk about the transcription in eukaryote. Now, when we talk about transcription in eukaryote, it is somewhat different than the prokaryotic transcription. So, let's study how it is different. If we want to study the different, first thing is that the RNA polymerase, which we have studied in prokaryotic transcription, it is only one type. Here, the RNA polymerases are present in nucleus and are of different type. So let's talk about the transcription in eukaryote. In eukaryote or in eukaryotic cell, three types of transcripting enzyme, three types of transcripting enzyme, that is RNA polymerases, are present in nucleus. And these three type of transcripting enzyme, that is RNA polymerase, are found in nucleus in addition to the RNA polymerase found in organelle. In organelle also it is found, but in nucleus these three types of RNA polymerase is found in. And this all three enzyme, if we talk, or three types of RNA polymerases, if we go talk, they are named as RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. If we talk about the RNA polymerase 1, it carry out 5.8s rRNA, 18s rRNA, and 28s rRNA synthesis. Whereas RNA polymerase 2, if we talk, it is involved in synthesis of hnRNA. hnRNA is heterogeneous nuclear RNA and mRNA synthesis. Then RNA polymerase 3 carry out a synthesis of tRNA, that is transfer RNA, small cytoplasmic RNA, 5s rRNA, small nuclear RNA. All this RNA are synthesized by RNA polymerase 3. So all these three RNA polymerases, if we talk, they have their specific function. Means this RNA polymerases show a clear-cut division of labor. So these RNA polymerases show clear-cut division of labor, because all these three different RNA polymerases have different function in case of the eukaryotic cell. Now first, the nascent RNA or the nascent RNA synthesized by RNA polymerase 2 is called hnRNA or primary transcript. Nascent means immature RNA. Immature RNA or unprocessed RNA, which is directly synthesized by the RNA polymerases, it is known as hnRNA, heterogeneous nuclear RNA, or primary transcript. Now, this primary transcript, it is modified by post transcriptional modification and form a processed mRNA. So this processed mRNA is formed from the hnRNA or primary transcript by the post-transcriptional modification. Now, first we are going to discuss how the process of transcription will occur, and later on we will discuss about the modification of hnRNA into nRNA by a post-transcriptional processing mechanism. So if we talk about the first, a transcription in eukaryote, and we are mainly talking about the mRNA transcription. So suppose that is 5 dash 2 3 dash, and this is 3 dash 2 5 dash, and that is DNA double helix. Now this DNA double helix, if we talk, if we talk about this DNA double helix, in this first unwinding occur and unwinding continues, and here RNA polymerase 2 carry out all steps like initiation, transcription, initiation, elongation, and termination. Now here, so if the initiation occur, initiation is 5 dash 2 3 dash, and this is a 3 dash to 5 dash, and initiation occur on this side. So this is the 5 dash to 3 dash end. So here mRNA initiation start, but it is not going to be mRNA, it is hnRNA. So this is initiation. Then elongation is also carried out by RNA polymerase 2. Here this initiation, if we talk, it is carried out by RNA polymerase 2. This is a particularly 3 dash to 5 dash. And here the RNA polymerase 2 carry out elongation in such a way the elongating hnRNA getting formed. So this is also what a hnRNA. Now after this hnRNA is formed and it is elongated, it is going to be terminated. So in termination step also this is DNA double helix. This is 3 dash to 5 dash and this is a hydrogen bond between them. And the hnRNA get terminated. Now this hnRNA is terminated in the presence of RNA polymerase 2 and now RNA polymerase 2 get also separated and this hnRNA is known as primary transcript. Now this hnRNA undergo post-transcriptional modification and this post-transcriptional modification going to form mRNA and this mRNA will have 7-methyl guanosine cap, poly A tail, and exonic sequences means coding sequences. Whereas hnRNA have intron, exon, intron, exon, intron, exon, intron, exon. Such coding, non-coding and non-coding sequences are there. Now we remember E is equal to exon, and exon is coding sequence, whereas I is intron, and intron is non-coding sequence. Okay, so this is the whole mRNA, but how what happens in this post-transcriptional modification, how this is formed, this matured mRNA is formed, that need to be studied. Okay, so now onwards we are going to talk about the post-transcriptional modification. Okay, so now when we talk about the post-transcriptional modification in eukaryote, which modify the hnRNA or heterogeneous nuclear RNA or primary transcript into the mRNA or functional mRNA. So in this, the post-transcriptional modification involve number one, splicing, number two, capping, and number three, tailing or polyadenylation. Okay, these three mechanism are there. Okay, and when we talk about this three mechanism, that is splicing, capping, and tailing is also known as polyadenylation. Then the first we are going to talk about a splicing. And when we talk about the first splicing, splicing is the removal of intron and joining of exon. So splicing, it is the process of removal of introns or non-coding sequences and joining of exon, that is coding sequences. So it is a removal of intron and joining of exon in defined order is known as splicing. And here introns are removed by small nuclear RNA small nuclear RNA and protein complex called small nuclear ribonucleoproteins or SNRPs. These small nucleoribonucleoproteins are mainly involved in the removal of this particular intron. And this presence of intron and exon is known as split gene arrangement. Split gene arrangement. Okay, so this intron exon are present in primary transcript or hnRNA, but they are not found in mRNA. So why? Because introns are removed. Now the question is how these introns are getting removed. So introns are removed like this. Suppose this is the pre-mRNA, or we can say hnRNA, and this hnRNA have an exon, intron, and exon. Now we want to remove this intron or non-coding sequence. The question is how it is getting removed. So to remove this snRNPs are used, and this snRNPs will form some coil-like structure of this particular intron, and this coil-like structure is known as spliceosome. Now this particular intron are removed in the lariat shape or a spliceosome form. It is removed, and snubs are also getting removed, and this particular mature RNA or mRNA have only intronic sequences. Okay, so this way the particularly introns are cut in between and removed by the snubs. Now after this we are going to talk about another mechanism, that is capping. Capping, if we talk, it is an unusual nucleotide that is known as methyl guanosine triphosphate. Okay, in capping what happens? That is the second mechanism. That unusual nucleotide methyl guanosine triphosphate is added at the 5' end of hnRNA. So see, suppose this is the hnRNA, and in hnRNA this is the cap of particularly 5-methyl guanosine is added here at the 5' end. Okay, and this cap binding protein add particularly this cap, then it is known as capping. Now this cap of 7-methyl guanosine triphosphate 7 methyl guanosine triphosphate is added to 5' end of hnRNA is added to 5' end of hnRNA by enzyme guanyl transferase, and this guanyl transferase add the cap of 7 methyl guanosine triphosphate at the 5' end of hnRNA. Now this cap is essential for formation of mRNA ribosome complex. So when translation will occur, this mRNA has to be attached with the ribosome. So to form a ribosome mRNA complex, this particularly cap. So mRNA and ribosome complex is formed by cap. And it is known that if cap is absent in mRNA, it is not identified by 18S rRNA of small subunit of ribosome, that is 40S, then translation does not occur. Because to translation to occur, there is a first step. We will study that first step is to bind with of mRNA with the small subunit of ribosome, that is 40S. And to bind with the 40S subunit, it has to make a bond. So let's see diagrammatically. Suppose this is the mRNA. And if mRNA has this particular cap, then it is going to form a bond with the 18S rRNA of 40S subunit of ribosome. And if it make a bond with this, then also this mRNA can be stabilized over the small subunit of ribosome. Okay. So such a way, this cap is essential for this particular process. And after splicing and capping two methods, the third processing method is tailing. Okay. And in this third tailing process, adenylate residues or adenine residues, there is a 200 to 300 are added at the 3' end in a template independent manner. Means template is not required, just there is a need of adding of this particular polyatyl or a 200 to 300 adenylate residue as the 3' end on a newly formed hnRNA. Okay, so here you can see this is a polyatyl, and this polyatyl is added here. Now we remember it is a fully processed hnRNA now called mRNA that transported out of the nucleus of the translation after this particular tailing mechanism. But we remember polyatyl is added by poly polymerase enzyme. Poly polymerase enzyme will add this particular 200 to 300 adenine residue as the at a 3' end. So it is also known as 3' tailing. Now after this 3 days tailing, the transcription is of post-transcription modification is over. Now we have a matured RNA. The matured RNA structure we have studied in the previous class in the particular mRNA. Okay, so this is all particular thing. Now we remember one important point of NCRT, that split gene arrangement, that is presence of intron and exon. If we talk, it is important. Why it is important? First, we should know that the split gene arrangement was discovered by Richard J. Roberts and Philip Shah. Richard J. Robert and Philip Shah. Now, the eukaryotic gene with exon and intron as intervening sequences is known as split gene arrangement. Okay. Now, this split gene arrangement or the presence of intron and exon, if we talk, it is considered that it represent probably an ancient feature of genome, and the presence of intron the presence of intron is reminiscent of antiquity, and the process of splicing. Reminiscence of antiquity means it is representing about some sequences of old origin. Okay. And the process of splicing represent the dominance of RNA world. Okay, dear students, so that is all about the what a transcription in case of the prokaryote, and that is all about the transcription in eukaryote, and that is all about the post-transcriptional modification in case of the eukaryote. I hope you understand. So the significance of subcomplex is now beginning to be understood. Split generation represent probability and sign feature of genome that we have discussed. RNA world in recent years, understanding of RNA and RNA independent processes in the living system has amazed more importance. Okay, so this is the short video which you can see here. There is a bacterial cell, and bacterial cell, this is the cell wall, this is central nucleoid material, and the cytoplasm and ribosomes are there. But nucleoid is important because if transcription is going to in nucleoid, the promoter is there, minus 35 sequence, minus 10 sequence is there. RNA polymerase will come along with the sigma factor. It will bind and initiate the process of transcription. Now, after binding, it will form a closed complex, and here Here, after forming a closed complex, it is promoter clearance will occur or initiate the particular RNA synthesis. Now, after initiation of RNA synthesis, further continuation, see, this is how RNA synthesis is initiated by the addition of ribonucleotides. Okay, so ribonucleotides are added are complementary to the template strand. Now, the elongation will continue like this, unwinding will continue, and the new ribonucleotide will added complementary to the template sequence. Ribosome is also getting added. We show that before completion of transcription, translation has initiated. And at the end, row factor will come and it will terminate the process of transcription. So this row factor dependent termination will occur. Okay. So this row dependent termination, you can see that is happening. Row factor is coming. It is binding here. It is terminating the whole process. So that is a prokaryotic transcription. It is shown in this particular video. It will help you to understand in a better way. Okay. Thank you, dear students.